The broadcast is now starting. All attendees are in listen-only mode.
Good morning to everyone joining us, and welcome to today's virtual event. This morning's talk is entitled Drug Development in Oncology in the Era of Precision Medicine. My name is Eric Falvey, and it is my pleasure to be your moderator for today's session. This morning's event will run for approximately 60 minutes, including time for a Q&A session with our speaker and panel. The session is designed to be interactive and works best when you're involved, so please feel free to submit questions and comments for our speaker by using the questions chat box, as seen here, and we'll tend to your questions during the Q&A session. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note the session will be recorded and made available to you for future viewing. This event is proudly hosted by IDDI. IDDI is an expert center in biostatistical and integrated eClinical services for pharmaceutical and biotechnology companies in several disease areas, especially in oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI is headquartered near Brussels, Belgium, with the U.S. Center of Operations in Raleigh, North Carolina. Now it is my pleasure to introduce you to this morning's speaker. Professor Christophe Tourneau is Senior Medical Oncologist at the Institut Curie and Professor of Medicine at Paris-Saclay University. He is heading the Department of Drug Development and Innovation, as well as the Head and Neck Clinic. Christophe is leading the early phase clinical trials program at the Institut Curie. Professor Tourneau was certified in medical oncology in 2005 and received his PhD in clinical epidemiology in 2007. His main interests are precision medicine, phase 1 clinical trials with a special attention at the methodology to conduct these trials, as well as head and neck oncology. He is the principal investigator of numerous phase 1 and 2 trials, as well as of clinical trials in head and neck oncology. Of note, he ran the first randomized precision medicine trial, SHIVA-1, that compared the efficacy of matched targeted therapy versus conventional chemotherapy in patients with advanced cancer. And now, it is my pleasure to hand the presentation over to Professor Christophe Tourneau. Christophe, it's all yours. Let me pass the controls. And there you go.
Hi, everyone, and thank you for the kind introduction. It's my pleasure to be with you today. So I'm not at home, but in the hospital, and this is the view we have behind me. So it's my pleasure to give an overview of how the drug development has moved in the past decades in the era of precision medicine with the molecular segmentation of cancer. So just some background to be sure we're all on the same page. And it is critical to separate two very different situations in oncology. The first one on the left is a patient with only a primary cancer without any distant metastasis. On the right, it is a patient with distant metastasis. And these two situations are completely different in terms not only of prognosis, but also of treatment. In the first case, the primary aim of any treatment is to cure our patients, and the most important treatments here are local treatments, including surgery and radiotherapy. We use sometimes drugs for a limited time, but these drugs won't be able to cure our patients, and surgeons and radiation oncologists will. Now we'll be able to improve the success rate of the overall strategy by using these drugs. When patients have distant metastasis, this is a completely different story, and here drugs play a very important role, and they are usually given for life. We're still using surgery and radiotherapy, but in a palliative intent. So that situation usually is not curable, but with some exceptions, such as germline tumors. And you all know Lance Armstrong, who had germline cancer with brain metastasis and who was cured from his cancer. So now the goal in that situation, unfortunately, is only to transform the disease into a chronic disease with patients receiving an effective treatment and having the best quality of life they can. So what has been the ancestral paradigm of drug development in oncology? Actually, it has started with chemotherapeutic agents that were discovered after the Second World War, and where they were tested in cancer cells and in preclinical models in specific cancer types, and then developed in specific cancer types. So we've got some chemotherapeutic agents that were effective in lung cancer overall, and others that might be effective in colon cancer, etc. Also, obviously, some chemotherapeutic agents might be effective in several cancer types. But this is how drugs have been developed per cancer type and in all comers. Actually, targeted therapies sometimes have also followed the same path. And if we look at anti-angiogenic agents or mTOR inhibitors, they have been developed exactly the same way in specific cancer types and in all comers without any molecular selection. And if we think about it, it has been the same for many immunotherapeutic agents, including anti-CTLA-4 or anti-PD-1 or PD-L1 agents. Most of these agents actually have been developed in specific cancer types without any molecular selection. Also, there are some biomarkers for some of them in some situations. And usually the classical path of drug development has been through phase 1, phase 2, and phase 3 clinical trial before market access. Phase 1 trials were usually non-randomized, and the primary endpoint here was safety in order to establish the recommended phase 2 dose based on the highest safe dose that we could administer to our patients, but also looking at PK and PD parameters and obviously preliminary efficacy. And I have to say, it's very rare to see a drug going into phase 2 if there has not been any efficacy in any patient. So then drug development went to phase 2, and here trials could be randomized or not. And the primary endpoint here would be preliminary efficacy using usually a surrogate endpoint of survival, such as overall response rate or progression-free survival. And obviously, safety was still a very important aspect to be sure that the recommended phase 2 dose actually is safe in a specific patient population with a specific cancer types. Whereas phase 1 clinical trials used to be performed in all comers in patients with any kind of cancer, phase 2 clinical trials are usually performed in patients with a specific cancer type and in a specific clinical setting, first line recurrent setting or second line, etc. And if results were convincing, then we went to a phase 3 clinical trials that are randomized. Here, the primary endpoint is the efficacy, and the gold standard endpoints are overall survival and quality of life. But here again, safety is important. So this is the ancestral paradigm of drug development that was applied for chemotherapeutic agents, but also for many targeted therapies and immunotherapies. Now things have changed a little bit in order to speed up drug development, and phase 2 clinical trials tend to disappear and are replaced by expansion cohorts performed in phase 1 clinical trials. So when the recommended phase 2 dose is being established, then within the same clinical trial, patients can be included in specific expansion cohort in specific cancer types and are treated with the recommended phase 2 dose. And this is a way to avoid setting up a new clinical trial. And usually, based on that drug would go into a phase 3 clinical trial without any phase 2. So this has become much more prevalent with time. So now we have also at the late 90s moved into a biomarker-driven drug development because some targets were found to be key in some cancer types, such as HER2 in some breast cancer patients or EGFR mutations in some lung cancer patients. And here some targeted therapies were developed in a specific cancer type, but with a molecular selection. And we have seen several targeted therapies being developed based on the molecular alteration, but usually, again, within a specific cancer type. And again, these drugs were developed the same way, moving from the classical phase 1 to 3 to phase 1 to 3 sometimes. So an example has been the crizotinib, which is a ALK inhibitor. ALK translocation are present in around 3% of lung cancer patients, and crizotinib has been developed in that setting. And here you see the result of the phase 1 clinical trial where the patient population was enriched with lung cancer patients harboring ALK translocation. And what you can see is that the response rate here is very high, and you see that when the scientific rationale is there and that the target is really a driver, then the efficacy of the drug is very high. So here, I guess I have a question for you. And given the results you saw just here, do you think that market access should have depend on the confirmation of a randomized trial against standard of care? And you can answer yes or no.
So thank you, Christophe. So we're now going to launch the poll question. Now, audience members, this is being done in real time, so your participation is very much encouraged and welcome. So please go ahead and cast your vote. And the question again is, given these results, do you think market access should depend on confirmation in a randomized trial against standard of care? Please select one of the following, yes or no. So we'll give just another few seconds for those of you who have not yet cast their vote. So please go ahead and do that now. Just a few more seconds. Okay, I think we've got enough coming in. So thank you everyone for participating to the poll question. We're now going to close the poll and share the results. And here are the results to the poll question. So 76% said yes to the question. Market access should depend on confirmation in a randomized trial against standard of care. So 76 were yes, and 24% of the audience said no to that. So thank you again, everyone, for participating to that poll question. And now back to Christophe. Christophe, what do you think of the results?
Yeah, thank you. So this is very interesting since I guess most of you, but not all of you, think that a randomized clinical trial would be important in that setting. Also, the response rate to that drug was very important. And a quarter of you didn't think it is important. So I would say I agree that randomization should be the gold standard to evaluate the efficacy of a new drug. Now we see that on the other side, actually the phase 3 was performed, and I can show you here the results. And indeed, crizotinib did much better than chemotherapy in ALK translocated lung cancer patients. And the fact is that they had to screen more than 3,000 patients in order to include around 300 or 400, I don't remember exactly. And that trial was very difficult to conduct. Also, it was feasible since lung cancer is a very prevalent cancer. So on one side, I agree this is gold standard. On the other side, we see that it is challenging to run a randomized trial when the alteration is only present in 3% of cancer patients. But we'll continue discussing this in a moment. So now it has appeared that some molecular alterations might be relevant not only in one cancer type, but in several cancer types. And in that setting, yeah, sorry guys, I'm not alone here. In that setting, some drugs could be developed not only in one cancer type, but in several cancer types. And this is why some new clinical trials were developed, namely basket trials. So these basket trials allow to evaluate a drug concomitantly in several tumor types. Now there are challenges. So the first one is when the incidence again of the alteration is low, and this can be very depressing for the investigator because they screen a lot of patients to only include a few, but also for patients who might be screened for a specific clinical trial, but the answer is that, oh, you do not harbor the alteration, so we cannot propose the drug to you. So it might be also difficult for the patients and also for the clinical research teams. And the other issue is that if you see nice results in terms of efficacy, then the challenge is how to make it to drug approval in maybe tissue agnostic way. And here we can discuss the NTRK inhibitors that have been developed against NTRK translocation that are present in only 0.3% of all cancer patients. And there are at least two drugs on the market now, larotrectinib and entrectinib. And if we look here at the results of the basket trial with larotrectinib, what we can see is that patients with various cancers harbored an NTRK translocation, but overall the response rate to this drug was very high, with most of patients actually responding to the drug. So now the question obviously is, if we follow the ancestral drug development paradigm, then we would like to have a randomized trial in every subgroup of cancer patients. And that obviously is not feasible, since even that basket trial took years to include around 100 patients. So if you want to run here a randomized trial in every cancer type, it will take decades actually. So another solution could be to run a randomized trial in all comers, all cancer types, provided patients have an NTRK fusion. But here again, that would mean to double the time of the clinical trial that was already with this single arm basket trial several years to include 100 patients. And the other aspect is that when you see such results with an efficacy that is high, is it relevant to give standard treatment to patients, knowing that the efficacy is so high with the drug? So for patients, in order to be able to propose them something, and especially not looking at a single alteration, especially when the single alteration has a very low prevalence, some clinical trials were developed that screened for multiple molecular alteration in a specific cancer type. And these trials are called umbrella trial. And they usually offer a treatment option in all patients with a specific cancer type, which is a good thing for these patients. They usually designed as parallel phase 2 clinical trials. Now there are several challenges again. First one being what is the prioritization in case a patient has several molecular alteration, and then if it is often the case, you might bias the result because of the treatment algorithm you've used. And again, some cohorts might not be filled because the alteration has a very low prevalence. Now, for patients, it's much more convenient because they know that participating in this umbrella trial, they will receive a drug eventually. So Upstream is an example of an umbrella trial run by the EORTC in head and neck squamous cell carcinoma patients who have recurrent or metastatic disease. And here you can see in green on the right that there are biomarker-driven cohorts based on molecular alterations. And these cohorts can be either single-arm phase 2 trials or randomized trials. And if patients harbor no molecular alteration, they are included in immunotherapy cohorts. But you see that doing this, there is a bias in the immunotherapy cohort since it does not reflect the overall patient population, but only the patient population without any molecular selection. So what we would like to do, obviously, is to be able to set up like a basket trial where we mix molecular targets, drugs, and cancer types. And what we would like is to be able to answer every subgroup of patients and to see whether in pancreatic cancer patients with that specific molecular alteration, the matched target therapy we have decided to give to this patient has efficacy. But obviously such a trial is impossible to set up because it would require probably tens of thousands of patients to be able to fill every single case here. So what has been performed is what I call algorithm testing trials. So these are trials that mix multiple cancer types, multiple molecular alterations, and multiple drugs. In fact, these trials have not the power to answer whether any of the drug has efficacy in any of the histologically and or molecularly defined subgroup of patients. But this kind of trial, they can only conclude on the efficiency of the treatment algorithm that has been used to allocate treatment. So here, this is really the treatment algorithm that is evaluated, but not any of the drugs that has been used in the clinical trial. And this is what we think is the precision medicine concept is given the molecular landscape of the cancer of a specific patient, what should be the treatment we should give to that specific patient. So there have been several clinical trials that have evaluated this concept of precision medicine, and the first one has been the one-off study published in the JCO in 2010. That has been really a pilot study. In that study, patients with any kind of cancer with a recurrent or metastatic disease could be included. Patients had to undergo a tumor biopsy, and then some molecular analysis were performed. So at that time, it was immunohistochemistry, FISH, and gene expression. And if a target was found, then a patient would receive match therapy. So in order to evaluate the efficacy of the strategy, they looked at the progression-free survival on match therapy, and they compared it to the progression-free survival on the last treatment patients had received. And what they found here is that 27% of patients had a PFS ratio exceeding 1.3. So in other terms, they had a PFS on match therapy that was at least 30% longer than the PFS on the last treatment. And if we look here at the patient on the right, that patient had a PFS of 2 months on last treatment and a PFS of 16 months on match therapy, suggesting that giving that drug to this patient was a good strategy. So now there are challenges when using the PFS ratio as an endpoint. The first one is that the assumption is that tumor growth is linear over time. So this is the red line you can see here. But some people will tell you that tumor growth in oncology is exponential because every cell divides. Some other will tell you that it is logarithmic because not only the surrounding cells divide, but at the end, if we consider that it takes like 20 to 30 years for cancer to develop in a patient, then over a very small period of time in the metastatic disease where we give treatments for months, sometimes for years, but usually for months, then we can make the assumption that tumor growth over this short period of time is linear. The second challenge is the assessment of PFS1 on the last treatment. And it is easy to understand that you need to check the timing of the efficacy assessment. Obviously, if during PFS2 patients are evaluated with imaging every 2 months, and let's imagine that during period A, PFS1 is being evaluated every 6 weeks, and let's imagine all patients progress on all treatments, then you will get a PFS ratio over 1.3 in all patients. Also, none of the treatments were effective at all. So you understand that the timing of the efficacy assessment is something that is critical and should be actually the same during the 2 periods. The second point is what is the method that has been used to assess the efficacy? Usually on PFS2, it is within a clinical trial, so RECIST criteria are being used, but on last prior therapy, it's usually standard of care, and we know that medical oncologists in routine, they do not use RECIST criteria, but they might change a treatment because of clinical progression or for any other reason. And again, if the methods are not the same for the 2 periods, then you might end up by biasing the results. So this is the second challenge. Another similar study than the Von Hoff study has been performed in France. That was the Moscato one, and they found here that 33% of patients had a PFS ratio exceeding 1.3. Here, they not only they looked at mutations using NGS and amplification using CGH. But here you also see a substantial proportion of patients with a PFS ratio that is below 0.7. So the PFS on PFS1 was longer than the PFS2, and this is the last challenge using that specific endpoint. The winter trial was an international effort, and again, it was a non-randomized trial, including patients with any kind of cancer. This patient had to undergo a tumor biopsy. They first looked at DNA alterations, and if they found any druggable molecular alteration, then patient would receive match therapy. And if they did not, they also looked at RNA, and using a treatment algorithm that they had developed, they would guide patients to specific treatment. And here again, the primary endpoint for both cohorts was a PFS ratio exceeding 1.5 in that case. So this trial was negative for its primary endpoint, with 22% of patients having a PFS ratio exceeding 1.3, but again with the same challenges I discussed earlier. In terms of randomized trials, the only one, to my knowledge, that has been published today is a Shiva one that we did perform at Curie Institute and in other centers in France, where we include patients with, again, any kind of cancer. All patients had to undergo tumor biopsy to be sure that the molecular profile would reflect the current disease. And we're looking for mutations, amplifications, deletions, but also expression of some using immunohistochemistry. So if patients had a druggable molecular alteration, according to a treatment algorithm we had set up beforehand, and I won't go through for the sake of time, but then these patients were randomized between match therapy according to our treatment algorithm and what they would have received if patients had not participated in the Shiva 1. So patients were then allowed to cross over disease progression. And actually, we didn't find any difference in terms of progression-free survival between the two arms. And that means that the treatment algorithm that we have used was not able to improve patients' outcome. Obviously, if we changed something in the treatment algorithm, the results might have been different. And indeed, in these treatment algorithm trials where we mixed cancer types, drugs, and alterations, what is the most important is the treatment algorithm per se. And that treatment algorithm has several features. The first one is a technology that is being used to identify molecular alterations. And you see here it's a now old study, but using 3 different machines, you see that they did not exactly find the same molecular, the same mutations using the 3 machines. So it's important not to change the machine during the trials. Otherwise, you might have sometimes different results. The same for the bioinformatic pipelines and the thresholds that are being used. Here is the same exome that has been assessed using 5 different bioinformatic pipelines. And what you see is that only 57% of alterations were common using these 5 bioinformatic pipelines. So here again, you need to set up the bioinformatic pipelines and the thresholds that you want to use beforehand, because if you change anything, it might change actually the result. The third point, and is probably the key point, is the matching between molecular alterations that are identified and the drugs that you're planning to use. And all these trials have so far used a unidimensional treatment algorithm. So what I mean with that is that we used to look at one target, and given that target, if it was altered, then we would give one specific targeted therapy. But what we know now is that probably the treatment algorithm should be multidimensional, and we should take into account coexisting molecular alterations, especially resistance mechanisms. As an example, we know that PI3K inhibitors might have efficacy in patients harboring a PI3K CA mutation, provided they do not have a coexisting KRAS mutation. So now, how to use these multidimensional treatment algorithms, and also how to set up them, that's another question. And I have to say, I don't know how to do this. The only thing I know is that I know that if we put all our results together from a precision medicine clinical trial, then we might end up having much more power to identify predictors of response to treatment. And this is something we are doing with Shiva 1, and we merge our result with the result of other clinical trials. And then the last point is what is the prioritization of molecular alterations if there are several. So over the last year, we found like every year a new paper publishing a scale for clinical actionability. And the last one here was the ESMO SCAT that tells you how important a molecular alteration is in a specific cancer type, depending on the clinical data available, but also the preclinical data available, what is the efficacy in other cancer types, etc. In fact, these scales actually are treatment algorithms, and they should be also evaluated like any other clinical trials, and they should be to validate them a randomized trial comparing drugs that have been given following the ESMO-SCAT as an example, and patients who received drugs without following the scale. So in a way, it's also a treatment algorithm, and we cannot consider that this is exactly what we should do since it has not been validated. So what are the perspectives? The perspectives, first, obviously, is to improve our treatment algorithms. We know in Shiva 1, there were some weaknesses in the drugs that we gave to patients. Before, we had no choice to give other drugs, right? And now there are novel drugs more specific for some molecular alterations. But not only drugs, but also to select the molecular alterations that are relevant for the trials. One way is also to use functional assays, so meaning not only looking at the molecular alterations, but also what is the function of the molecular alteration. And here it's a very small study, and you cannot conclude anything from this, but we went back to the Shiva 01 clinical trial, and retrospectively and in a blinded way, they looked that a small company, they did perform a functional assay on the molecular alterations we had identified, and they did say whether they would predict efficacy or not of the drugs that we gave to our patients. And we got nice curves here. Also, it's a very small sample size, obviously, but it suggests that maybe the functional assay they use is relevant beyond what we did with our treatment algorithm to select treatments. Second thing is maybe to get rid of pathology was not completely the right way, at least in some instances. And what we have decided in a clinical trial that we just started last month, the PIVOT trial, that we decided to work in a specific histological subtype, namely squamous cell carcinoma of different origins. And we know that these squamous cell carcinomas, they share molecular alterations. So we decided to set up a basket trial with different cohorts of patients with head and neck, lung, cervical, penile, vulva, anal squamous cell carcinoma. And here we decided to evaluate one specific drug combination, which is immunotherapy with pembrolizumab that we know has efficacy in some patients, around 15 to 20% of patients. And we would like to improve that response rate. And here we added an epi drug, vorinostat, which is an HDAC inhibitor, because it has been shown that the epigenetic modulation might improve the efficacy of immunotherapy. And a bunch of translational studies are associated in that study. Perspectives are also based on molecular tumor board, and it has become much more common now to have a molecular tumor board in institutions where patients are treated. And this is what we do at Curie Institute every week. that medical oncologists can discuss their patients with recurrent or metastatic cancer. And here we would see whether it is relevant to have a comprehensive molecular profiling. And one of the key aspects is that the patient is in good general condition. What we don't want is to perform comprehensive molecular profiling and saying eventually to the patient that he is not eligible in that specific clinical trial because he does not fill the inclusion criteria. So then we choose what kind of analysis we should do on what kind of sample. Is it archived sample, or do we need to perform biopsy? Do we have frozen tissue, FFPE, or maybe on ctDNA? And all this is discussed for every patient, depending also on what clinical trials are open in the institution and within our partners. And then everything goes to bioinformatics, and we discuss the results within our molecular tumor board and make a proposition of inclusion in a biomarker-driven clinical trial if we have one. So it's a way to spare tissue and time because we're looking for multiple molecular alteration in the same analysis, and also a good thing for patients, obviously, because once we have identified molecular alteration, then we would look for the relevant clinical trial for them and not reverse way. Now, perspectives are also to overcome the challenge of small patient populations. As I mentioned earlier, for molecular alterations with a very low incidence such as NTRK fusions, it's really tricky to run a randomized trial. And here we are working on new methods to address these situations. The first, and I mentioned it in several clinical trials that were run, the PFS ratio, but with challenges. We set up the SHIVA-02 trial with that methodology using the PFS ratio, but here we decided that the PFS 1 and 2 should be assessed exactly the same way. So with exactly the same timing for efficacy assessment every 2 months, but also using the RECIST criteria on both periods. So that SHIVA-02 clinical trial is running now, so more than 250 patients were included, and hopefully we'll be able to present results next year or within 2 years. Another perspective is real-world data. So far, it has been, I guess, not a very good success because I think the quality of the data that is being entered in these huge databases are not high enough. So what we need is to have real-world databases with very high quality, with many patients, with a heterogeneous patient population, but most importantly, to have patients' characteristics, molecular alterations, because obviously the problem of small patient population is linked to the molecular segmentation of cancer, and also treatment outcomes. And this is key. We need to know exactly what response to treatments patients had, and this cannot be done using just softwares taking information from charts. So if you want to get very good data, so you need to have people entering the data in a CRF, like in a clinical trial, and this is the only way to do it. So we are working on this with the Wayfinder project collaboration with Roche, and hopefully we'll be able to get high quality data. And then once you have all this data, you might within that database randomize patients treated with a specific treatment based on specific molecular alterations, and maybe to adjust for confounding factors, and to be able to conclude on the efficacy of a specific drug in a specific subgroup of patients. So with that, I would like to conclude. So the point is that most of treatments in oncology are not biomarker driven. chemotherapy, obviously, but also many targeted, so-called targeted therapies and many immunotherapeutic agents. It is true that biomarker-driven treatments usually demonstrate a very high efficacy, but they benefit only a small proportion of cancer patients. The concept of precision medicine has become a reality with the tissue-agnostic approval of some drugs by the FDA, including NTRK inhibitors, but also anti-PD-1 agents in patients with MSI-high tumors, independent of primary tumor location, and even more recently, patients with a high mutational burden. Also, we could discuss on that. I think there is not a single way to develop drugs, so it is not like we went from the ancestral drug development paradigm to a new one. So the ancestral drug development paradigm is still very important and key and applies for most of drugs actually. But for novel drugs such as NTRK inhibitors or RET inhibitor based on specific molecular alterations, we need to find novel ways to develop these drugs with a challenge of randomization that is tricky in these cases. And at the end, I would conclude by saying I think that a comprehensive molecular profiling should be proposed to every patient with recurrent or metastatic cancer, but only provided that patients have access to biomarker-driven drugs and early phase clinical trials with matched therapies. There is no way we should have a comprehensive molecular profiling if we are not able to propose anything to this patient thereafter. It would be the worst thing to do. So with that, I would like to finish with a question for you, and relate to the identification of biomarkers. So for you, what is the optimal time to try identifying biomarkers during the drug development process, during the preclinical phase, during phase 1 trials, during randomized trials?
Thank you, Christophe. So yeah, we're going to launch the last poll question now. So again, audience, it's done in real time. So thanks again for your participation. So the question again is, what is the optimal time to try identifying biomarkers during the drug development process? And please select one of the following. It's during the preclinical phase, or during phase 1, or the other selection is during randomized trials. So please go ahead and cast your vote, and we'll give it just a few more seconds to wrap up here. And the results are coming in. So, okay, thank you everyone for participating to that poll question. We can now close that poll and share the results. And here are the results. So the optimal time to try identifying biomarkers, 48% said during the preclinical phase, and a close runner-up second, 44% said during phase 1, and at the very last, trailing at 8% was during randomized trials. So Christophe, yeah, just to get your thoughts on this results to this question.
Yeah, I guess it's very interesting again how you answered that question, and I guess the optimal situation would be in the preclinical phase. I agree with you, because then it's much easier to develop the drugs such as trastuzumab in HER2 amplified breast cancer patients. But I think we should also take every situation to try to identify biomarkers. So what we see is that in biomarker-driven trials, the response rate, and this has been published in several meta-analyses of phase 1 and phase 2 clinical trials, the efficacy is much higher. So if the biomarker is being identified during the phase 1, such as ALK translocation, it was the case, this is good. And also during randomized trials, I mean the EGFR, the KRAS mutation in colorectal cancer for patients treated with cetuximab. Also, cetuximab was initially given to all patients independent of any biomarker. eventually discovering that KRAS mutation as a biomarker of resistance was something that was very important for our patients in order to avoid to give them a drug that is not effective. So it's not the optimal setting, I agree, but I think this is also a situation where we should be able to look at our data and work with pharma to go back to the data in order to try to identify these biomarkers. So with that, I would like to thank my team at Curie Institute in the Department of Drug Development and Innovation. It's a pleasure to work with them, and I would like to thank you all for your attention, and I would be very happy to take any question. Thank you.
Well, thank you very much, Christophe, for that excellent presentation. I hope everyone has enjoyed that. So now we can begin with the Q&A portion to wrap up the webinar. For the Q&A, we've invited two members of IDDI to join the panel, Dr. Everardo Saad and Dr. Elizabeth Coart. And I just want to remind the audience members that you can continue sending in your questions right now using the questions window. I've already received some questions, so I'll start with those. Looking at the question box, so let's begin. Precision medicine consists of developing drugs that have very high efficacy in very small patient population. This raises two questions. Is the model economically sustainable? The cost of drugs in precision medicine is quite high. Two, is there still room for developing marginally effective drugs for all comers? So I'm not sure which of the the panel would like to take that one. But
yeah, I think both questions are very good, and I think it's sustainable. So I'm not an expert in the field to develop drugs in small subgroup of patients. Because the point is that usually these drugs are very effective, and then they are given for a long time for these patients. I know there were some papers published on that, and comparing the relevance of developing drugs in biomarker-driven situations or in all comers. And at the end, I remember a paper in JCO, but a few years ago already, and they showed that from a financial perspective, it was sustainable because these drugs worked in many more patients and for a very long time. And so at the end, it was sustainable. Now, I'm not an expert, but I would say from now a patient perspective, it's probably a very good way to go. Is there still room for drugs in all comers with a marginal effect? I think to fight cancer, we need to go in all directions. It's better to have very effective drugs in small subgroup of patients, but I mean, if we can improve also the outcome of these patients step by step, and this is what has been done with chemotherapy, for example, in metastatic colorectal cancer. And we went from an overall survival of around 6 to 12 months to now 3 years. So adding some targeted therapies, I agree, but also new drugs such as oxaliplatin, irinotecan, and at the end, a few months plus a few months, it makes several months, and it improves us the life of our patients. So I think there is still room for this.
Well, thank you, Christophe. Everardo or else, did you have anything you'd like to add to that, or can we get to the next question? Anything you'd like to add to that one, or?
No, not from my side, Eric.
Okay. Everardo, I think you're on mute, but...
Yeah, I'm still muted.
Sorry about that. I was just saying, let's go to the next question, because I see that there are others that are also interesting, and Christophe's answer was quite good.
Okay, excellent. So next one, why is the assumption of linear tumor growth required for the PFS ratio to be used?
Yeah, I think if it's not the case, so if tumor growth is not linear, so you might either overestimate the efficacy of match therapy or underestimate. So depending of whether the tumor growth is exponential or logarithmic. Now, I agree from a clinical perspective. When a patient had a treatment that had efficacy for 4 months and then another one that had efficacy for a year, then you don't need to go into explanation like how is tumor growth over time, logarithmic or exponential or linear. The patient is happy with the second drug because it had efficacy for a longer time than the previous one. So I think from a methodological point of view, we need to ask ourselves this question. But if we only look at a clinician perspective and a patient perspective, then I agree that question is probably not that important.
Okay, thank you, Christophe. Here's one. It's actually, well, it's kind of a combined two questions, but let's get to it. So does it have to be decided at the time of the study start whether the drug will be evaluated primarily in the overall population, the biomarker-based subgroup, or both? And what is, statistically speaking, the most powerful approach?
So I guess from a statistical point of view, it is more relevant to decide beforehand in which patient population this will be evaluated and how many patients are needed to enter the question. Now it's not always feasible, so sometimes there are hypotheses raising up during the clinical trial on a potential biomarker, and in many instances actually we don't know, and it's more like a fishing expedition. So we have tumor biopsies and we have some clues, but we don't know exactly which of the biomarkers beforehand will be relevant, and in that situation it might be difficult to have a sample size calculation, but just an overall sample size calculation saying, okay, if we think that the biomarker would be present in, I don't know, 20% of patients, then we need that amount of patients, but we never know beforehand because some molecular alteration might be present in 20% of cases, but sometimes in 40%, and it might depend. So it's very hard to say, but the ideal situation is to have an idea about the specific biomarker, about the prevalence, and to also calculate the sample size based on that.
Yeah, thank you, Christophe. Everardo, else, did you want to weigh in on that one?
No, maybe else could
yeah.
I don't know if you had
Yeah, I completely agree, of course, with Christophe that there's the clinical situation that we, as statisticians, we need to be able to deal with. So we can't change the amount of information that's already available at the time that the study starts. But there are methods, like you say, Christophe, to adapt the trial while it's ongoing. There are methods to start the trial only in the biomarker subgroup. So it's really, I think, a question about a good collaboration between the clinician or the clinical team and the statisticians to put the right statistical methodology given the knowledge we have on the biomarker and the drug. Yeah.
I fully agree. Thanks.
Thank you, Els. Well, looking at the time, I'm sorry, guys, we're going to have to wrap up here. Yeah, I guess we're going to have to. Well, we've officially reached the end of the question and answer portion of today's session. Please note for the audience, if we couldn't attend to your questions, we'll be sure to follow up with you after this presentation. Now, if you do have any further questions, please direct them to me at my email as shown on this this slide here, eric.falvey@iddi.com. I wanted to thank everyone for participating to today's virtual breakfast event. You will be receiving a follow-up email from us with access to the recorded archive for this event. Also, a survey window will be popping up on your screen when we close this session. So again, your participation is very much appreciated as this helps us to improve future webinars that we'll be doing. And a final thank you to our speaker, Professor Tourneau, for that very insightful presentation. So we hope you found this webinar informative. It's really been my pleasure to be your moderator this morning. On behalf of our team, we thank you for joining us. And as a special thank you for joining, all participants will be receiving a voucher for some handmade luxury Belgian chocolates. So hope you enjoy that. So I'm Eric Falvey. Until next time, please take care and bye for now.